Hello everyone. So today we are starting this program. So thanks Ensembl EBI for accepting our invitation. I am requesting Professor Sudeshna ma'am as a HOD to make the introductory speech. Ma'am, over to you. Ma'am, you are muted. Ma'am, you are muted.
Can you hear me?
Yes, ma'am.
Okay. So, welcome to Microbiome Department. As I have already told, that bioinformatics is a very important area of life sciences, which acts as a bridge in between wet lab and dry lab. So far, these bioinformatics have been included in our CBCS UG curriculum, PG curriculum, as well as in PhD coursework. So we know that the data derived from the wet lab can be banked, can be used, can be fetched for further analysis for our research purpose. So far, when we are working for bioinformatics, we use this Ensembl, that genome browser, that server, which actually helps us to know about the bioinformatics database. Not only bioinformatics database, but also many of the tools which are governed by the Ensembl also helps us to make a bridge to coordinate our research data, to analyze our research data. So far in our bioinformatics class, we become able to explain the biomolecule, the characteristics, the functioning of the biomolecules. It might be DNA, it might be protein, it might be RNA, whatever. So far, the biomolecules as well as gene expression could be analyzed by bioinformatics tools. So once again, we are very much honored that directly from Ensembl, this workshop has been organized. And once again, thank you, Dattatreya as well as Bikram Dhara, who are coordinating this webinar. So I think our scholars as well as our faculty members will be definitely benefited from this workshop. Thank you so much.
Thank you so much, ma'am, for your support all through this workshop. Now, I'd like to ask Dr. for
This
conducting
workshop
the.
is under the scheme DBT Star College. So where Professor Arup Kumar Mitro is actually coordinator of this, convener of this DBT Star College, and Dr. Mahashita Mishra Ghosh is departmental convener, coordinator. So far, I also want to acknowledge them and Professor Mitro, who has took this initiative for this workshop. Thank you.
Thank you, ma'am, for your introductory speech. Now I am requesting Ben sir and Louis ma'am for starting the program. Thank you, everyone.
Thank you very much. And thank you very much for the kind introduction. And I also wanted to extend our gratitude to everyone over in St. Xavier's who's been instrumental in organizing the workshop and making sure that everything is in place ready for today. So I wanted to start by introducing myself. So my name is Ben Moore. I'm one of the members of the Ensembl outreach team. And within the larger Ensembl organization, our team is responsible for delivering courses and workshops to introduce people to the Ensembl genome browser and teach them how to use it in their day-to-day work. So I'm going to be delivering some of the sessions in the course today, tomorrow, and on Thursday. And I'm joined by my colleague, Louise. She's also on the call today, and she'll be delivering some of the other modules over the course of the next few days. We're both going to be here for the whole workshop. So that means that we can each help each other answering the questions and providing support for you in the chat box and when you have questions in the exercise sessions, for example. So I just wanted to start by giving you an overview of the different topics that we're going to cover in the course over the next 3 days. So what I understand and the information that I have about you from the survey that you filled in before the course is that we have a wide range of levels of experience of using Ensembl. Some of you have never come across Ensembl before or used it in your work. Some of you have already seen it and used it perhaps a little bit in your research. The other thing that I noticed is that there's a very wide range of interests of all of you people who are joining us on the workshop. So some of you are studying human genes and genomes and diseases. Some of you are studying microbiomes. Some of you are studying bacteria, and some of you are even studying mangroves and lots of a range of different species and levels of interest. So what we wanted to do in the course is give you an introduction to a number of different types of data and a number of different tools that you have available in the Ensembl Genome Browser. So today I'm going to start off by introducing you to the main principles of the Genome Browser, how the pages are laid out and the different types of data that are there. Then we're going to move on and begin looking at the Ensembl genes. So we're going to think about how Ensembl takes the genome sequences of different species and annotates them with the genes and the transcripts. Tomorrow, Louise is going to be introducing you to variation data in Ensembl. So thinking about those variants that exist within the genome that can be linked with phenotypes and diseases, for example. And then she'll also move on and talk to you about the regulation of gene expression. So thinking about how Ensembl annotates genomes with features that have the ability to control the level of gene expression. On the final day, on Thursday, we have 3 smaller modules. So firstly, Louise is going to talk to you about the comparative genomics data that we have in Ensembl. So thinking about how we compare the genes and the genome sequences between species to understand homology and conserved regions. And then finally, Louise will talk to you about the Biomart data export tool that we have in the Ensembl browser. So all through the week, we're going to be using the web-based browser for finding information about individual genes and individual features. But the Biomart tool allows us to begin looking at how we can export data on a larger scale. At the very end of Thursday's session, I'm going to give a short wrap-up session that talks to you about how you can visualize your own data in the Ensembl Genome Browser. And then finally, we'll move on to having a wrap-up and an opportunity for you to have feedback as well, to give feedback. So each of these modules has quite a particular structure. So we'll start off with a presentation. We'll think about the background to the data or what the tool that we're discussing is. And we'll think about how Ensembl processes or produces that data. Then we'll move on to having a hands-on demonstration. So the idea in this section is that I will lead a walkthrough of that particular section of the browser, looking at that data and looking at the different features that are on the pages. Hopefully, if you are able to have a couple of different windows open on your computer, you can then also follow along with me as well. So you can begin to use Ensembl for yourself and begin navigating around and learning. You don't have to follow along. We're very happy for you to watch if you think that that's the way that you're going to learn the best. Then finally, we'll finish with an exercise session in each of the modules. And these just give you an opportunity to begin practicing using Ensembl for yourself and exploring the different pages that we've just been looking at in the demo. So the exercises are not a test. They're not going to be marked in any way or graded. It's just an opportunity for you to explore and learn by yourself. What we have in the exercise sessions is a range of different exercises that cover different species and different areas of interest. So please do spend your time at the beginning of each exercise session and just read through the exercises, look to see if there are any exercises that are particularly interesting to you, in which case you can have a go at those first. And then if you have time, you can move on to the others as well. If you have your own species of interest or your own gene of interest that's not covered in the exercises that we have for that module, then please feel free to leave the exercises completely aside, and you can even just begin exploring, using that time to explore your own gene or your own species of interest. So the first thing that I wanted to do was to show you where you can find all of the materials that we have for the course over this week. So we have a very specific page where we have all of the materials, and it's hosted through our training pages. So this is the web address that you need to visit at the top here, training.ensembl.org. So I'm going to go there now, and I'll actually just show you how you can find the training materials. So if you open up a tab in your internet browser and type training.ensembl.org, I'll put that in the chat box for you, so you can click on the link if you need to. This is not the Ensembl Genome browser. This is the training pages. So this is where we have information about all of the workshops that we're delivering. So what you can actually do is you can find the link in the bottom left-hand corner here with this magnifying glass. So if you click on this link that says find materials from training, it will take you to a calendar of our training events. So here you can see we have our event here listed, the St. Xavier's College browser workshop. So we can click on the link here, and this will take us to a page that has all of the information about the course. So over on the right-hand side, we have a PDF of the presentation slides. So you can please feel free to download this and keep this for reference in the future. Underneath the presentation, we also have a link for what we call the living document. So if you click on this link now, this will take you to a page, a Google document, which is available for everyone to access and to edit. If you scroll down, you can see that we have some information about the course. You can see this all listed here. We have some links for the slides and the materials. We'll also add the URL of the workshop recording after the workshop as well. Some links for further resources. And then at the bottom, we have a section for your questions. So the idea is that this is a living document that everyone in the course can add to and to edit. So if you have questions throughout the course, then please feel free to come to the living document and ask your question here. So if you had a question, maybe I had a question in the course, I could just put my name in here and I could type my question. And then obviously myself and Louise will monitor this document and we'll be able to put our answer in here. The idea is that then everyone can see the different questions and the different answers. And this document is available in the future. So you can always come back here and see the questions that were asked and the answers that Louise and I provided. So it sort of becomes like a knowledge base and a reference source that you can use in the future as well. Good. So hopefully you all have access to the documents. If you have more shorter technical questions, then you can also feel free to use the Zoom chat box as well. Going back to the documents, the final thing that I wanted to show you here is this section called demos and exercises at the bottom. So what you'll notice is that we have the titles of all of our different modules that we're going to cover in the course this week. And so if you click on the first one, for example, this is the first module that we're going to cover in a moment. You can see that once you click on the title of the module, you open up a section where you're able to view the demo. So the demo is the walkthrough that we're going to do. So these are the annotated screenshots and instructions. So obviously you can follow along live with me, but you can also come back here and use this as a reference to remember the steps that we took to go through the browser. The other section here are the exercises. So here we can see that we have a number of different exercises. These are the questions, which you can obviously spend some time answering. And then if you get stuck, we have a text answer and also a video answer that you can watch as well that shows you how we got the answers for those questions. So this is these are the materials that we're going to be using all through the course this week. And we'll direct you back here and give you the links if you get lost as well. Good. So as I said, during the exercise session, you can spend some time having a go at the exercises yourself. But if you get stuck, Louise and I will always be here to help you with your questions. So you can use the Zoom chat box if you have short questions or if you have trouble with the audio or the visual, for example. But the best place to ask questions will be the living document. This means that you can provide links and screenshots and put them all in that Google document, and it just becomes easier to make longer answers and longer questions. Obviously, sometimes in the Zoom chat, if lots of people are asking questions, it can get a bit messy and difficult to see where the question and the answer is. Good. So over the course of this week, there's a number of different learning objectives that we have. So firstly, we want to introduce you to what Ensembl is. We're going to think about the different types of data that are available. And we want to show you how you can navigate around the Ensembl browser, the website itself, to get those different data. So you might want to look at specific data and you want to retrieve them for your work. So we want to show you how to navigate around Ensembl to get that data that you need. And then finally, we want to show you where to go for help and documentation. So we only have 3 days together. So we're not actually going to be able to cover everything that's available in the Ensembl browser. We're mainly going to be giving you an introduction to lots of different aspects of it. So if you're using Ensembl in the future, you are likely going to need further help and learning and documentation. So we're going to show you where you can go for that as well. So before we start the workshop itself, I wanted to ask you a couple of questions just to get your input and to start the discussions. So we actually have a live poll that's organized through a platform called Slido. So if you have a phone or another device available to you, or even just another window in your web browser, you can either scan this QR code in the top left, or you can go to the website here. So you can type Slido. You can type slido.com into your website, into your internet browser, and then type the code here, 9430481. So then if you access Slido through that code, you'll be able to answer our first poll. So the question is, have you used Ensembl before? So I'd just be interested to know, of all of you who are attending the course today, who's had a chance to use Ensembl in their work before and might have some experience with it, and those of you who have not. So I'll just give you 1 or 2 minutes to answer the poll. Okay, so I can see that some of you have been able to access Slido. I think some of you are having trouble with Slido, so I think you might have even written your answers in the chat box as well, in the Zoom chat box. But I can see already that we have 13% of people saying yes, that they've had a chance of using Ensembl before, but 88% of people have said no, and that includes even 4 people in the chat box who haven't used Ensembl before. So obviously, as I said, that's absolutely not a problem at all. This is an introductory workshop, so hopefully what Louise and I are going to show you is the different things that you can do with Ensembl. I think when people start to use Ensembl, they're not even aware of all the different types of data that they can access. So that's what we really want to do, is introduce you to the different things that you can do. For those of you that have used Ensembl before, hopefully we'll still be able to show you some of the other data or tools that you haven't been able to explore before, and really show you how you can integrate that into your work. Good. So we just have one more question that I want to ask before we move on to the next, to the first presentation. Is, even if you haven't used Ensembl before, what do you think Ensembl is? So what we're going to do here is build a word cloud. So I just want you to use the same approach. So go to slido.com and enter this code, so 9430481, and then you'll be able to enter 1 or 2 keywords that just describe what you think Ensembl is. So what do you use Ensembl for? What data does it have? What is it useful for? So I'll just give you a couple of minutes to answer that poll now. I can see a couple of you are already writing. If you're having trouble with Slido, you can also put your answers in the Zoom chat box as well. Okay, so I think 1 or 2 of you are still typing your answers. I can see it in the Zoom chat box as well. So the thing that obviously is jumping out to me is the words that I think most people have used here: genome browser. So I can also see genome search browser, search genomes of interest, genome browser, and data analysis. So I think a lot of you, even if you haven't used Ensembl, have a good idea and a grasp of the fact that Ensembl is a genome browser. So the main data type that we have is the genomes themselves. So we have the genomes of lots of different species, and we annotate those genomes with different information. So genes and transcripts and variants and regulatory features. So it seems like lots of you already have a good grasp and a good concept of the main data types that we have in Ensembl. So it looks like you already have a good understanding. Hopefully we can just teach you more depth and more information about Ensembl in the workshop this week. Good. So let's move on to the first module. So what I want to do is introduce you to the Ensembl project and some of the different concepts that we have when we're using the Ensembl browser. So genome browsers are very important tools. So Ensembl is not the only genome browser that's available. There are other genome browsers that host similar data, but Ensembl is one project that's based in the EBI in the UK. And well, the first organism actually to have its genome sequenced was a bacteriophage. So back in 1977, Fred Sanger and his team in Cambridge sequenced a bacteriophage, the genome of a bacteriophage, which was only 5 kilobases in size. So by today's sequencing standards, that's obviously a really short genome, but that technology laid the foundations for more and more complex organisms to have their genome sequenced, until in the early 2000s, we had the publication of the first draft of the human genome sequence. So you can see that the human genome is around 3 gigabases in size, so 3 billion base pairs. And that obviously was a major landmark in sequencing genomes and making that data available. But even since then, we have the advent of next generation sequencing technologies. So what that means is that more and more complex genomes are being sequenced. So we have, for example, a number of different plants that have really large genomes. They get their genomes sequenced to quite a high quality. And we also have projects that are aiming to sequence lots of different species all over the planet. There's the Darwin Tree of Life project, for example, that is aiming to sequence all of the eukaryotic species in the UK. And then we have the Vertebrate Genomes project as well, for example, aiming to sequence all of the vertebrate species on the planet as well. So the idea is
can I
that,
ask a question?
of course, yes.
You just mentioned this vertebrate genome database just now.
Yeah.
So
Yeah.
there was this one database which you mentioned right before that. Can you please say it again? I missed it.
Yeah, so it's called the Darwin Tree of Life project.
Okay. And what's the purpose of it?
It's a collaborative effort across a number of different institutes and research groups to sequence all of the genomes of the eukaryotes that are indigenous to the UK, the United Kingdom.
Thank you.
So the idea is that there's a number of different stages of this project. So there's obviously sample collection, sequencing. So there's the Sanger Institute, for example, in Cambridge is going to be responsible for doing lots of the different sequencing. And then Ensembl comes in at the end of this project, where we are, it's our role to host and to annotate those genomes and make them available through the genome browser. The only reason why I really mention these large projects, though, is to try and demonstrate that what we have at the moment in the field of genomics is this explosion of data. So where the technology is improved so much, and even the cost has come down dramatically, it means that genomes are being sequenced at a very, very high rate. So lots of different genomes are having, lots of different species are having their genome sequenced, but also lots of even individuals within a single species. So the 1000 Genomes Project is a good example of a project that tried to sequence and identify variants from thousands of different individuals around in different populations around the world. So we have this accumulation of genetic data, genome sequences and variants, for example. So this is what Ensembl tries to do. We try and take all of this data that looks a bit like this. So we take this raw sequence data and we annotate it with lots of different data types. So the first thing to mention, I sort of alluded to it just now, but it's important to remember that Ensembl is not a sequencing project. So Ensembl doesn't do any of the sequencing of the different genomes that are available. So we rely on different research groups and collaborations to sequence the genomes of different species, and then to deposit those genomes into public archives. So you might have heard of the archives such as GenBank and ENA. So these are nucleotide sequence archives. So once the genome sequences are available in those publicly available archives, Ensembl can take the genome sequences, like this is just a fragment, for example, you can see on the screen, and then we annotate that with the genes and the transcripts, the variation data, and then we perform all of the comparative genomics analyses as well, for example. So this is why Ensembl and other genome browsers are really useful, because when we just look at the genome sequence, like you can see on the screen here, this doesn't really mean anything to anybody. It's just a string of A's, T's, G's, and C's. So with the annotation, Ensembl tries to make the genome sequences as useful as possible to the different scientists around the world who need to use it. So to do this, Ensembl has 4 main types of data. So the first type of data, we're going to cover each of these in the modules in the course this week. So the first type is the genomic assemblies. So as I said, Ensembl doesn't do the genome sequencing ourselves. We take the genomes that have been deposited into the archives, but then we annotate those genomes with genes using an automated gene annotation system that I'm going to describe to you in the module later this afternoon. The second type of data is the variation data. So this can include both small and large scale sequence variants with things like allele frequencies and phenotype associations as well, where that's available. The third type of data is the comparative genomics. So this is where we can compare the genes and the genomes from one species to another to predict things like orthologs and paralogs and highly conserved regions. And then the fourth type of data is the regulation data. So this is where Ensembl annotates features that regulate gene expression, such as promoters and enhancers. And we're going to cover each of these topics through the course this week. So to draw these 4 branches of data together, Ensembl has a number of features. So The first is the gene build. So this is what we call, this is another name for the gene annotation. So we have a gene build or gene annotation for over 300 different species in Ensembl now. And then we also have the variation, comparative genomics, and regulation data display as well. One of the other things that you can do is you can display your own data in Ensembl. So Ensembl is not a submission database. So we don't accept people submitting their data to include in the genome browser in general. But if you want to visualize your own data alongside the publicly available data, you can do that. And I'm going to teach you how to do that on Thursday. We also have a number of tools for data processing and analysis. So an example of this would be the VEP, the Variant Effect Predictor, which is a tool for analyzing your own variation data sets. And we're going to teach you how to use that tomorrow. Finally, we have these different data access methods. So we've got the Biomart data export tool for exporting tables of data from Ensembl. And then we also have programmatic access as well. So what we're going to cover in the course today is how to access the data through the website. But if you are familiar with programming languages such as Python or R, you can retrieve the data programmatically through our REST API as well. If you're interested in this, we're not going to have time to cover it in this course, but please let us know and we can point you towards some documentation so you can explore that for yourself. The best thing about all of this is that it's completely open source. So all of the data that we have is completely free for you to take and use in your work. So as I said, Ensembl has over 300 different species. This is just an example of the species tree that we have in Ensembl. What you can see if you look very closely is that we obviously have human and then the other great apes. And then we also have a large number of other primates and placental mammals as well. We try and cover a wide range of species, including agriculturally important species, conservationally important species, and evolutionarily important species as well. As well as placental mammals, we also have marsupials and monotremes. So if you're studying mammalian evolution, that's obviously an important groups to study. And then we also have a large number of other birds, reptiles, fish, and amphibians as well. So if you had, I don't know, you wouldn't have probably noticed this because of the size of the image on the previous slide. But if you looked at all of those species, that what you'd have noticed is that all of the species that we have in Ensembl are vertebrate species. So Ensembl is a project that aims to present the genomes and the gene annotation for vertebrate species. So you can see here, human, mouse, and zebrafish are our three most commonly accessed species in Ensembl. We do have some representative non-vertebrates there as well. So we just have the yeast, Saccharomyces cerevisiae, the nematode worm, C. elegans, and then we also have Drosophila melanogaster, the fruit fly as well. So if you want to access the vertebrate annotation, then you need to use our main website, www.ensembl.org. However, I know that a lot of you are studying non-vertebrate species, so plants and microbes as well. So what we actually have is a sister project called Ensembl Genomes, where you can find a similar data, but for non-vertebrate species. So we have taxa-specific pages for bacteria, fungi, protists, metazoa, and plants as well. So you can see here, this is the separate website. So this is www.ensemblegenomes.org. So I always think it's a bit confusing with the names because we have the Ensembl Genome Browser and the Ensembl Genomes Genome Browser. But all you have to do is remember, am I studying a vertebrate? If so, you go to ensembl.org. Or if you're studying a non-vertebrate, you need to go to ensemblegenomes.org. What we're going to do in the course is have a look at both of these different pages side by side, and we'll show you the similarities and the differences. But the pages are generally laid out in pretty much the same way. So everything that you learn in the workshop today, you can apply from one to the other. One of the important things to remember when you're using Ensembl and Ensembl genomes is that we work with a release cycle. So hopefully you appreciate that these genome sequences and genomic data is being continually produced by different research groups and added to databases and archives around the world. So what Ensembl has to do is we release a new version every 2, 3, or 4 months to make sure that we include the latest data in our databases. So at the moment, for example, we have Ensembl release 106, which is our current version of Ensembl, and that was released in April of this year. What we're currently doing is we're looking for new genome assemblies for different species. We're updating our gene annotation. We're reperforming our comparative analyses, updating our interfaces and our other data sets. And then we're going to package that all up and we're going to release it as Ensembl 107. So that's actually going to be released in a couple of weeks. So in July, we'll have Ensembl 107. And what this means is that everything is very traceable. So if you're looking at a specific gene in a specific species in Ensembl, the annotation or the available data might slightly change between 2 releases. But what we do is we have we make the archives available. So if you're looking at Ensembl today, for example, you know you're using Ensembl 106. And if you come back in a year's time and you want to see the data as you saw it today in this workshop, you can come back and visit the Ensembl 106 archive. And I'll show you how to do that. So before we start the first demo, I want to think about the concept of genomes and genome assemblies. It's the main type of data that we have in Ensembl. So we can think of the word genome, and we can sort of broadly describe this as the DNA within a cell. So this could be, if we think of an analogy, this might be the globe, Earth. What we have in Ensembl, though, is actually what we call a genome assembly. So a genome assembly is a representation of the genome based on the sequencing that we've obviously performed in the lab. So this can be what we think of as a map of the world. So what we can, again, to extend the analogy, we can think of a map as having gaps and errors. So if we think back to maybe the 16th or 17th centuries, the technology was not so good, and the maps that we had of the Earth, of the globe, were very inaccurate. They had gaps and they had errors. But as the technology improved, the representation of the Earth became much more true, much more accurate about to what the globe actually is like. And therefore the technology, so now the maps that we have are pretty much a faithful representation of the Earth. So that's what we have in the genome. So we can see that the genome assembly is obviously put together as a map of the genome using the sequences that we have from the sequencing machines. But as the technology improves, sometimes the gaps and the errors are filled in and corrected so that we have as faithful representation of the genome as we can. And it's the genome assemblies that we have in Ensembl that you're going to be looking at. So what is a genome assembly? So obviously you have the genomic sequence of the DNA in the cell, and it's split often into chromosomes. So for vertebrate species at least. So what we actually have, and other eukaryotes. So at the moment, there's no technology that can sequence a whole chromosome in one go. So what we actually have to do is we fragment the genome into short fragments, and then we sequence those short fragments of DNA. So you can see here we have these short fragments that are sequenced, and then what we do is we use alignment software in the computer to align these reads to match up where they overlap. So you can see here, for example, there are 4 different reads that all agree when we align them that at this position there is a T. So the genome assembly is the inferred sequence from where the sequenced reads overlap and align. So you can see here there's a T, and then a C, and then a C. Now this cartoon that I have here obviously makes the process look very simple, but actually what you'll find is that because you because we're dealing with such a large amount of data, what we actually might find is that there are regions of the genome that are not represented. So there might be gaps. Maybe these two reads didn't get sequenced here, so there would be a gap in the genome here. You can also have some errors, and you can have some problems with the alignment if there's repetitive regions as well. So this is not a perfect process. As I said when we were talking about the map, it's not a perfect process. So there are improvements that are continually made as the technology improves. So this slide is slightly out of date. It sort of is based on older technology, but it still does represent the way that the human genome was sequenced in the early 2000s. So what happened is that these fragments that are generated from the genomic sequence, the DNA, these are cloned into what we call bacterial artificial chromosomes, called BACs. So these are plasmids that are sequenced, and then the clones from the BACs are assembled together with overlaps to form what we call the tile path. So this representation of these 4 blocks, this is the tile path, and these are the individual BAC clones. So this is one BAC clone, and this is another BAC clone. Then the overlaps that you can see here are trimmed to give us what we call the contigs. So the contigs are represented as these light and dark blue blocks. So you can see that this is a contig that comes from this clone, and this is a contig that comes from this clone. A run of contigs together with no gaps is called a scaffold. So this is one scaffold, and this is another scaffold. And then the final step is to use genetic maps to place these scaffolds on the chromosome. So we have to work out where these scaffolds exist relative to one another and in what orientation as well. So this is the final step in producing the genome assembly. And finally, it's important to remember when we're looking at genomes for different species, is that they might have been produced using DNA from a single individual or a number of different individuals. So if we take the human genome as an example, the human genome is actually a hybrid genome that's made up of chunks, so the contigs that have been sequenced from a number of different individuals. So it's a composite or a hybrid genome. So we can see here, for example, this contig comes from this individual, and this contig comes from this individual, and this contig comes from this individual. Now, a single chromosome is going to have many more than 4 contigs. This is just an example here, but the idea is to show you that the contigs actually come from people that are going to have obviously particular ethnicities and phenotypes and characteristics. So what we have is just a single hybrid genome that represents every human on the planet in this case. This is going to become really important when we're thinking about variation data tomorrow. So the final thing I wanted to mention to you before we start the first demo is that we have different versions of genome assemblies. So as I mentioned, as the technology improves over time, we can make corrections and improvements to the genome assemblies. And so what generally happens is that new versions of the genomes are released and made publicly available. So if we take human as an example again, we have GRCH38. This is the most current and up-to-date version of the human genome assembly, and it's what you'll find if you access www.ensembl.org. However, before GRCH38, there was what we call GRCH37. So this is the previous version of the human genome assembly. This was slightly poorer quality, so there's increased gap length. And then now the annotation is not regularly updated either. So we do have an archive that's dedicated to GRCH37. You can see the archive here. But many people are now moving towards GRCH38 as the main reference genome assembly that they're using. Good. So I'm just looking to see if there are any questions at the moment. Obviously, there was one question that was asked aloud in the presentation. I can't see any questions in the chat box. Are there any questions at this stage before we move on to the first demo?
So can I ask a question?
Of course, yeah.
So I want to know which is the most popularly used aligner software used for these purposes?
Alignment software.
Yeah, the most popularly used aligner software used for content generation.
Oh, okay. Wow. I'm not actually. I'd have to look into this actually. The best thing to do would probably be to ask the question in the living document. Can you type it in there? And then I'll have to look into it and research some of the methodologies and get back to you.
Okay, sure. Thank you.
No problem. Okay, perfect. So if there are no further questions, what we're going to do is move on to our first demo. So we're going to look at the Ensembl home pages and see how we can find information about the different species and the genome assemblies in Ensembl and in Ensembl genomes as well. So if you want to follow along, you can obviously open up a new tab in your genome browser, but you can also come to the training pages and find the demo here in the species and genome assemblies section. I'm going to open a new tab in my browser and I'm going to type www.ensembl.org. So when you come here, you can see that you have arrived at the Ensembl homepage. There's quite a lot of information here, so I'm just going to spend a few minutes just explaining the different links that are on the page. So at the very top, you can see we have this blue navigation bar. This will appear at the top of every page in Ensembl, so you can always use the links here to continue searching or to navigate to your next place that you need to. So the logo is a link to come back to the home page, so you can always come back to the Ensembl homepage and start your searching again. We've also got links to our BLAST and our BLACK tools. The VEP tool as well, we have a link there. But we also have a range of other tools that we're going to explore throughout this course as well. So you can find all of our tools through the tools link here. Then we have a link for Biomart, which Louise is going to talk to you about on Thursday. Then we also have links to give you information about downloading large data sets from Ensembl, and then also our help and documentation as well. So I'm going to click on the help and docs now, just to show you how that's arranged. So if you click on help and docs, this will take you to all of our documentation pages. So here you can see we have a section that teaches you about the 4 different data types. So we have Ensembl annotation, variation data, comparative genomics, and regulation data. So you can read a lot of background information there as well if you're interested in those different data types. But then we also have a glossary of terms and frequently asked questions, these sorts of things as well. So if you ever get stuck and lost, then this is a good place to come and begin exploring Ensembl for yourself as well. I'm now going to click on the logo to go back to the home page. Beneath the blue search bar, beneath the blue navigation bar at the top, you can see this search bar underneath. So this search bar is your main portal into the whole of Ensembl. So this search bar, you can search for things like gene names, gene IDs, you can search for variants, you can search for phenotypes, and we're going to use this search box throughout the course this week to search for all of our different data that we need. Over on the right-hand side, you can see we have information about the release number. So we can see that we're currently in Ensembl release 106. So that's the current version of Ensembl, and obviously, we're having Ensembl 107 released next month. So if you come back later on in July, you'll see that this was Ensembl 107. That will be our current latest version. Further down the page, you can see we have information about all of the different species that we have represented in Ensembl. So you can see we just have some quick links to some commonly accessed species. But over on the left-hand side, you can either drop down the list here to find all the different species. However, because we have so many species, I find this list quite difficult to use. What I prefer to do is click on the link here. So the link will take you to a table that shows you all of the Ensembl species. So if you click on the link here, view list of all species, this takes you to a page where you can see that we have a table with all of the species. It's obviously a very large table, and you can explore it in your own time. What you can do, though, is you can search for your own species of interest using the filter in the top right-hand corner here. So if I just begin typing Homo, for example, to search for Homo sapiens, you can see that my table is now filtered to show human. So we've got human, the scientific name, the taxonomy ID for human, and then we also have the Ensembl assembly name, so GRCH38. And then you can see that this is the accession number. So this is the unique ID that that represents the GRCH38 genome assembly. Now, this accession number is quite important because if you're using different tools and different databases online, when you're looking at the genomes and different sequences, the genome assembly name might be called different things in different databases. So, for example, you might see it called HG38, so for human genome 38, but they'll always reference this ID number. So as long as you can see that the accession number or this ID number is the same, it means that you're using the same data when you go from one genome browser to another or to different tools online that you're using. So if you want to find out more information about your species, you can click on the common name in the table. And this will take you to a species-specific homepage. So now you can see we can have information all about human, if human is our species of interest. And then we have you can see the assembly name. You can see we've got a search bar at the top here. So you can use this search bar to search for human data specifically. Then underneath we have blocks with the different data types. So we've got the gene annotation data, comparative genomics, regulation, and variation as well. In the top left-hand corner, we have information about the genome assembly itself. So you can see again the assembly name and the ID. Underneath, we can see a drop-down box that shows us the other assemblies that are available for this species. So we know that GRCH38 is the current most up-to-date version of the human genome assembly. But if you open the drop-down menu, you can see the other genome assemblies that are available. So we have GRCH37 available for human. And also, even before then, there was a genome called NCBI36. So these are the versions of the human genome assembly that are available. And if you want to use them, you can select the one from the list and then click go to go to the archive. But we'll just stick with the GRCH38 genome for our example. The final thing I want to show you here is if you click on more information and statistics, this will take you to a page that has the background information about the genome assembly, who produced it and when, and the technologies that were used. So you can see here, for example, the assembly, background information on the top left here. Then we have further down information about the gene annotation methodology. And then over on the right-hand side, we have some statistics. So you can see, for example, the name of the assembly and the accession number. You can see the base pairs. So this is the number of base pairs, the length of the genome sequence. Then we have, for example, the dates of when the genome was annotated with the genes, for example. And then tables underneath that give us the number of genes that are annotated across the whole genome. So, for example, on the main primary assembly, without any alternative sequences, there are 20,471 protein coding genes. And then you can also see there's a number of non-coding genes and pseudogenes are annotated as well. So these are maybe not so important when you're doing your research, but this just gives us a general overview of the statistics for the genome and the gene annotation across the whole species. So before we start our first exercises, I wanted to show you the Ensembl genomes pages. So if you're studying a non-vertebrate. So if you open up a new tab in your internet browser, you can type www.ensemblgenomes.org. So that will take you to our Ensembl Genomes pages. I can put the link in the chat box for you. So Ensembl Genomes, as I said, is a place where you can find information about non-vertebrate species. So it's divided into the different taxonomic groups. So we have one special case here for the SARS-CoV-2 genome sequence. So Ensembl does not have viral genomes, with the exception of the coronavirus SARS-CoV-2 genome. But then apart from that, we also have Ensembl plants, Ensembl metazoa, Ensembl protists, Ensembl fungi, and Ensembl bacteria. So if, for example, you're interested in plants, then you can click on the link here to go to the Ensembl plants pages. So you'll see that it's laid out in a very, very similar way. You can see it's green though to tell you that you're in the Ensembl plants division. You've got the search bar, you've got some information about the different archives on the right-hand side, and then you can also click through to find a list of all of the different plant species. Just as we did in Ensembl, you can search for your species of interest and go to the species-specific home pages to get information about them. The only thing that I will do is I'll also show you the Ensembl bacteria pages. So if you go to www.ensemblgenomes.org and this time go to bacteria, you can see that these pages are laid out slightly differently. So we also have the navigation bar and we have the search bar at the top, but the search bar is laid out slightly differently because we have so many bacterial genomes in Ensembl. I think we have around 40,000 species and 40,000 genomes, that it becomes very difficult to search. So what we can actually do is you can begin typing species into the genome box here. And then the box will auto-complete. So you can actually now begin looking at all of the different species that have this, your keywords that you've searched here. So for example, I just began typing Escherichia for E. coli. So now you can begin seeing all of the different E. coli strains that we have in Ensembl. So these are all separate genomes that have their own IDs. Maybe they're different strains from different isolates, for example. If you want to find out more, then you can click on the species, and that will take you to the species-specific homepage. And again, you can search and find out more information about this genome assembly. Good. Okay. So that brings me to the end of the first demo. So what I want to do now is just give you an opportunity to have a go at the first set of exercises. So if you come back to the training pages, I'm going to put that in the chat box for you here. You can find a section that is called Species and Genome Assemblies. And then you can see our exercises that we have. So we have one exercise on panda, one exercise on zebrafish, one exercise on mosquitoes, and then one exercise on bacteria as well. So as I said, there's quite a lot of exercises. We don't expect you to complete them all in the time that we have together. But just the wise thing to do, the best thing to do is to look through the exercises and pick the one or two that are the most relevant or interesting to you and your work, and have a go at those exercises and then move on to the others if you have time. So I think we'll have around maybe 10 minutes to have a go at this first set of exercises. And then we'll come back together, see if there are any questions, and then we'll think about moving on to the next module. So it's now, well, it's 10:30 here in the UK. So I think that means it's around 3 o'clock in the afternoon there in India. So we'll come back together around 3:10 to go over any questions that you have about these exercises. Good. I will mute myself, but please do get our attention if you have any questions during the exercises. Okay, good. Okay, so if there are no further questions,
then
yes.
Hello.
Hi, Bikram.
Yeah, actually, I wanted to know that is it mandatory to fill both the text answer and video answer? So that
Sorry?
was my question. Actually, in the exercises,
Yeah.
we have to fill the text answer and video answer both, or should we just fill the text answer part?
So you don't have to. I mean, no. So the answers that we have there in the materials, these are just to help you if you get stuck. So you don't have to look at the. I mean, you can just make a note of your answers in a document or on a pad of paper that you have with you.
I thought yes.
Okay, yeah. So these are just to support you if you get stuck and offer you some like next steps and some help. So as I said, the exercises are not graded. We don't have to submit the exercise.
Yeah. Please carry on. Yeah.
Okay. Yeah, no problem. Okay, good. So let's move on to the next module then. So in this next module, what we want to do is actually begin exploring the genomes themselves. So we're going to look at what we call the region in detail view. So this is going to help us to explore specific genomic regions within our genomes of interest. So we're going to use human as an example in this demo. So we're going to look at a region of the human genome that's on human chromosome 4. So you can see these are the coordinates that we have here. So we're on human chromosome 4, and then we have our start coordinate here and our end coordinate here. So what we're going to do is we're going to search for these genomic coordinates and then look at the genome of this region and look at all of the annotation that we can see there. So if we go from the presentation, I'm going to open up my tab with the Ensembl genome browser, and I'm going to go back to the home page by clicking on the logo, so I can just begin to search from the home page. So if you want to search, you can search using the blue search box on the home page. So the first thing that we have to do is select our species from the drop-down menu. So I'm going to firstly select human, and then into the search box itself, I'm going to type in the genomic coordinates of the region that I want to search. So I'm going to start with the chromosome number, which in this case is chromosome 4. And then we need a colon to indicate that we are now going to give our start coordinate. So then our start coordinate is 122.868.000. And then we have a hyphen, just a small dash, and then our end coordinate, which is 122.946.000. I'm going to paste these coordinates into the Zoom chat box, so you can also copy and paste that if you want to for your search, if you're following along. So you can see that we have quite a specific format here. So we have the chromosome number, then a colon, then the start coordinate, then a hyphen, then the end coordinate. And we don't have any other gaps or punctuation either. Good. So when you've typed in the coordinates, you can click go. And this will take you to what we call the location tab. So you can see here that in the blue bar at the top, we've begun to open up different tabs. So at the moment, we just searched for a genomic location. So we are now in the location tab. As you click on more features and explore more things like more genes and more transcripts, then we're going to open up more tabs in this blue bar at the top. So we have a location tab for exploring genomic locations. We'll have a gene tab for exploring different genes, and we'll have a variant tab for variants. So the different types of features have got different types of tab. So underneath the tab, you can see the menu on the left-hand side. So you can see these are all of the different views for exploring this, this genomic location. At the moment, we are exploring this page called Region in Detail that you can see is highlighted here in the menu. What you can also see is that we have a section for comparative genomics, genetic variation, and you can even use the links here to explore this genomic region in other genome browsers such as UCSC and NCBI. So these are other genome browsers that are hosted by other research groups. Underneath the menu, you can see these 5 blue buttons. Now, these are really important and they're going to help us manipulate the data on the page. So the first button is called Configure this page. So we're going to use this in a moment, but just to tell you now, what this button does is it helps us to add or remove data from the main images on the page. So just by default, we preload a small amount of data on this page, but there's lots of data available, and we can use the options here to add more data tracks to our view. The next button is called Custom Tracks. So we're going to explore this option on Thursday when we want to visualize our own data sets. So with the Custom Tracks button, you can upload your own data to visualize alongside all of the publicly available data in Ensembl. The next button is called Export Data, and we're actually going to click on this now to explore this in more detail. So if you click on Export Data, you'll open up a pop-up window that allows you to export the data from this genomic region. So your first set of options are to do with the output file format. So by default, we've selected FASTA sequence. So if you want to get the sequence of this genomic region, you can select FASTA, it's already selected here. But we also have a number of different file formats here as well. So if you want to retrieve, for example, a list of gene features, you can use the GTF format, for example. So the different format that you select will determine the different data type that you want to get. So I don't expect you, or I don't think that you will be familiar with all of these different file formats. But if you know that you want to get the sequence, for example, then you know that you need to get FASTA. Then you have options for selecting the different parameters and the different options, which you can obviously change depending on what you need. And then you can click Next. From here, you're given an option to either view the sequence in a web browser, so in HTML, or you can download the FASTA file as a text file or a compressed text file. So you can download these if you want to, or you can view the sequence in the web. So you can click on HTML, and then you can see, for example, here we have the FASTA sequence of this genomic region. Obviously, this is not that useful at this stage, but you might want to take this sequence and use it in some other downstream tools, such as a primer designing tool or some alignment tools that you have outside of Ensembl. So this is how you can export the sequence and other data from this region. Good. So back to the tab that we had with our location tab. So I'm just going to click on the tick in the top right-hand corner of the pop-up window now, just to close that and go back to the region in detail view that we had. The last two options over on the left-hand side are just to share and to bookmark the page. So you can do that if you need to. On the main part of the page, now we're going to actually begin exploring the data that you can see here. So you can see that the data is divided into these 3 separate images, and each one is more detailed than the last. So the top image is what we call the chromosomal overview. So this is just, you can see the whole of human chromosome 4. You can see the P arm here. This is the centromere, and then this is the Q arm. The first thing to notice is this red box. So you can see the red box on my image here. This is our region of interest. So this is the genomic region that we searched for with the coordinates when we started. Outside of this red box, you can see that we also have these other red and green highlighted regions. These are what we call assembly exceptions. You might not always be interested in them. So for example, in our region that we have searched for, these assembly exceptions are not in this region, so it doesn't concern us in this case. But if you want to find out more information about the assembly exceptions, you can hover your mouse until they turn into this finger icon and then click on them. And you can see that you can find more information about what assembly exceptions are, and you can compare it with the reference. So it's what essentially assembly exceptions are there patches and haplotype sequences that are added extra to the main reference genome sequence. So we have what we call the primary assembly, which is just the main sequence. But then perhaps if there's a gap or an error in the original sequence, what we do is we lay an extra bit of sequence on top called a patch that will just correct any gaps or errors in the original primary assembly. So rather than changing the original, we just provide a patch or a sequence that overlays this gap or error and corrects it. And that's what you can see is highlighted here. So these are the different patch and haplotype sequences. Good. Going further down the page, we can see that we now have a more detailed image, and this is called the 1 megabase overview. So you can see the scale at the top here. This is 1 megabase, and you can also see the red box. So this again is my region of interest, and you can see that as we zoom in, we're becoming more and more focused on our red box. This is our region of interest. So this is our start coordinate here, and this is our end coordinate. The first thing to notice is that the data is arranged into these tracks. So what we have is the tracks that just represent different data types. So the first track to find and to familiarize yourself with is the contigs track. So as we described in the first presentation, the contigs is the representation of the genome itself. So the contigs that are arranged end to end form a continuous sequence that is the double-stranded DNA sequence in human here. So here we can see, for example, this blue bar, that's the light and dark blue, this is the representation of the genome itself. And so this is 5' to 3' on the forward strand going left to right. And then this is the reverse strand going back in this direction. So this blue bar represents the genome sequence itself. And then the data in all of the other tracks is aligned to this reference genome sequence in the contigs track. So you can see here we have a track called genes. So you can see now that all of the genes are represented as these colored blocks, and each block is a single gene. So we have this big block here that's like a gold color. This is the SPAT-A5 gene. Then we have here this gold block here. This is the NUD-T6 gene. So at the moment, we can just see that the genes are represented as these colored blocks. The color, you can find out what the color means in the legend at the bottom, but we're going to think about this in the next module today. As we scroll further down the page, we can see that we now have what we call the region in detail view. So this is the genomic region that we searched for. This is our red box. So this is our start coordinate over on the left-hand side here. And this is our end coordinate over on the right-hand side. Again, the data is arranged into these tracks, and you can view them in more detail. So again, you can see the contigs track here. So in fact, our view is just looking at one single contig here. But obviously, we know that if we were to zoom out, we would see that more contigs extend obviously away from our region of interest. And then we also have these tracks above and below the contigs. What you can notice is that the genes track is now split into two. And rather than blocks, the genes are actually represented as their individual transcripts. So in Ensembl, we use the terminology transcript, but you might also refer to them as isoforms or splice variants. These are basically essentially the different transcript sequences that are transcribed from the single gene. We know that a single gene can have many different transcripts that are expressed in different times in different tissues. So what we can see here is that in this track, we have the 3 transcripts of the FGF2 gene. And then further downstream here, we have the different transcripts of the SPAT-A5 gene. Now this track is showing us the genes and the transcripts that are on the forward strand. So these are all forward stranded genes and transcripts. Underneath, you can see that this is the NUD-T6 gene and its individual transcripts. These are all on the reverse strand. So this is the 5' end of these transcripts, and the transcription is going in this direction, right to left on the screen, on the reverse direction. Good. If you're interested in these features, you can actually click on them. So if you click on them, you'll open up a pop-up window that will give you more information about the gene and its location, and further links as well, which we're going to explore later on in the demo today. Good. Further down, you can see that there's other data types that are all added by default, and then there's a legend at the bottom as well. This can help you work out what the different colors of the blocks means. But we'll obviously discuss all of these things as we're looking at the individual data types. If you scroll further up the page, what I want to do now is click on the blue configure this page button on the left-hand side. So this, as I said, will allow us to add more data to our view. So you can click on configure this page, and this will open up a pop-up window that shows us all of the tracks that we can add to the region in detail view. So in the main part of the pop-up window, you can see all of the active tracks. So these are the tracks that are all turned on by default. But on the left-hand side, you can see the subcategories of the different tracks that we have available. So, for example, we have tracks that show us information about the sequence and assembly. You can see tracks that show us information about genes and transcripts. You can see tracks for mRNA and protein alignments. You can see variation data tracks. And what you can actually even notice here is that most of the tracks are turned off, and you can tell that they're off because the box here is empty. So these are the ones that are turned on, the tracks that have an icon in the box here. So the idea here is really that there are so many tracks, and we're never going to have enough time to show you each of the tracks and to talk about the data that's in them. What I would advise you to do is spend the time in the demo and in the exercises today looking through the different tracks and thinking about what tracks are going to show you the data that's important and relevant and interesting for your work. Obviously, everyone is going to have different areas of interest and will want to add different tracks. So what we can do is we can explore some tracks here. So we're already in the variation section here by clicking in variation on the left. So if we want to turn a track on, maybe this one here, to turn a track on, all you do is you click on the box, and then from the next pop-up window, you can choose the formatting option. So maybe just normal in this example, and you can now see this track is turned on. So you can just spend your time looking through the tracks and turning them on by clicking on the box. If you're not sure what a track is going to show you, you can click on the information icon, and it will just give you a short description about what the data in that track is, or where it comes from, or what it represents. If we scroll back up to the top of this pop-up window, there is a filter, a search function in the top left-hand corner. So if you're not sure where your track is, but you have maybe a descriptive word or phrase that you want to search for, maybe if you want to search for proteins from UniProt, you can just type UniProt, and then you'll find all of the tracks that show you protein alignments from the UniProt database. So maybe I can turn on the mammalian protein alignments by clicking on the box and selecting the format. So these are just two examples. I've just added two tracks to show you how it works. You can obviously spend some time looking through the different tracks. When you're happy, you can save and close by clicking on the tick in the top right-hand corner. And you can see that the page at the bottom, this region in detail view, is now reloading with the data that I just added. So now you can see that as well as the data that we saw before, we now have these other tracks that are added. So these are the mammalian protein alignments from UniProt with these gold blocks. So you can see here, this is for the forward strand. And then we also have the mammalian protein alignments from UniProt to the reverse strand. Further down, you can see that we also have this track from the LOVD database. So this is the other track that we added. If you want to further manipulate the data on this page, you've got a number of options. So you can move the tracks relative to one another. So if I was interested in the data in this track, but I wanted to see it in the context of the tracks further up the page, I can just hover my mouse until it turns into this arrow icon, and then click and drag, and I can move the track up the page. So now I can perhaps look at the data in this track side by side with the data in these tracks further up the page. So you can move them all and arrange them how you need them. If you want to do, if there's more actions that are available for each track, so if you click on the track name, this will open up a pop-up window where you can get firstly information, or you can change the formatting of the track, you can share the track, or you can highlight the track or delete the track. So these are just different options, quick links to do some quick things with changing the format of the track. So if you don't want this track to be visible anymore, you can just turn it off by clicking on the cross, and then it will remove it from the view for you. Good. The other options that I just wanted to quickly show you are the dragging and box options in the top right-hand corner. So you've got this double-headed arrow and the dotted box options. So if you click on the dotted box, this means that you can select a region of interest. So maybe if we are looking at our region and we think, okay, this is an interesting exon of this FGF2 gene that I want to explore, you can draw a box around your feature of interest. And then when you let go of the mouse, you're given an option to either mark the region or to jump to the region. So if I click jump to region, the page will reload and it will recenter it on that box that I just drew. So you can use the box drawing function to zoom in on a region of interest if you want to see it in more detail. So you can see here now I've zoomed in on this region. If you click on the double-headed arrow, this just changes the functionality of the mouse. So now when I click and drag, I can actually move upstream and downstream. So because Ensembl is an online browser, every time you click and drag, the page has to reload. So it can take sometimes a couple of minutes to reload the page. So this is not the best option for sort of exploring large regions of the genome, but if you just want to move around and explore the upstream and downstream region, then this is the best option here. Good. The final thing that I want to show you is the options in the top left-hand corner of the image. So there's a few different options for sharing and resizing the image. You can also export the image. So if you click on this icon, you can, in the pop-up window, you can save your image as in a number of different formats for presentations or posters or web pages, for example. I won't download anything now, but you can obviously do this for in your own time if you need to. And then the other options here are to reset the configuration and to reset the track order. So using these buttons, you can go back to the default view. So you can reorder the tracks back to their default order. And when you click reset configuration, you'll turn off all the tracks that you turned on. So you'll go back to the default track settings. Good. So that's everything that I wanted to show you. What I want to do now is give you maybe 15 minutes to have a go at the exercises for this session. And then we're going to take a break. So if you go to the training materials, you can open the next module called Exploring Genomic Regions. And here, again, you'll find some exercises. So we have the exercises for exploring a genomic region in human. We have an exercise looking at CRISPR sites. If you're designing guide RNAs for CRISPR, this might be quite an exciting exercise for you to do. Exploring assembly exceptions, and then we have a plants region, using rice as an example, and then also another bacterial exercise as well. So please spend the next 15 minutes having a go at these exercises. And then, yeah, in 15 minutes, we're going to come back together and see if we have any questions. And then we're going to take a short break before our next module. So if you want to take we're going to start again in 25 minutes. So that will be 11:30 here in the UK, which translates to, I think, 4 o'clock there in India. So if you make sure that you're back here for 4 p.m., we're going to spend the last hour doing our going over the last module. So please spend 15 minutes doing exercises. Then we're going to take a 10-minute break. We're going to start again at 11:30 here in the UK, which is 4 p.m. India Standard Time. Please do ask any questions in the living document or the chat box if you have any questions. Hello, everyone. Welcome back to the final hour of our session today for the Ensembl Genome Browser workshop. Hopefully you're able to just take a 10-minute break just now and are refreshed and ready for our final module. Before we move on to our final module, I wanted to ask you if you had any questions from the previous set of exercises or the demonstration, or if there's anything that you're stuck on or anything that you want to have clarified. Good. So if there are no further questions, let's move on to our next module. So what I want to do now is think about the genes and the transcripts. So we've already had a look at the genes and transcripts in the location tab. But what I want to do in this module is think in more detail about how the genes and their transcripts are annotated and what information we can get about them through the Ensembl Genome Browser. So when you are looking at the genes in the Ensembl Genome Browser, you're actually looking at the output of two independent methods, gene annotation methods. So the first is an automated gene annotation pipeline, and the second is manual annotation by a group of curators. So for every species in Ensembl, you can see here just an example, like these are the species that we have in the Ensembl Genome Browser, the vertebrates. All of the genes that are annotated across these species are annotated by an automated pipeline. So there's no human curation involved, but we've designed and developed a pipeline that aligns evidence to the genome and uses it to predict the genes. For a small number of other species, so human, mouse, rat, and zebrafish, there is a group of curators that have moved along the genome, annotating the genes manually using a range of other evidence types. So long read sequence data, publication data, mass spec data. So lots of different data types are being used to annotate the genes for these 4 species. So for these 4 species, for human, mouse, rat, and zebrafish, the genes that you actually are looking at in Ensembl is the product of these 2 independent methods being combined together. So for human, for example, the automated pipeline predicts a set of genes using a range of biological evidence. And then the curators also produce a gene set using the evidence that they use in their annotation. And then we bring the output of those 2 methods together to produce the final gene set. Now, obviously, in many cases, the methodologies actually agree, and they come up with the same gene annotation model and location and length and structure. In some cases, there's a discrepancy between the 2, in which case we represent the full list of all of the annotation. For a number of species that do not have the manual annotation, then obviously the genes that you're viewing are just the output of the automated gene annotation pipeline by itself. So let's think of these two methods in a bit more detail. So firstly, the automated gene annotation. As I said, it's a pipeline that makes predictions of the gene models across the genomes of all the species in Ensembl based on experimental data. So these are the types of data that the automated pipeline integrates into its annotation. So we firstly include nucleotide sequences that come from the INSDCs, the International Nucleoside Sequence Database Consortium, which is made up of 3 databases. So ENA, GenBank, and DDBJ. Together, these 3 databases form the INSDC, and they have nucleotide sequences, so things like cDNA sequences, ESTs, express sequence tags, and RNA-seq data as well. So for different tissues, there's RNA-seq data sets available. So the automated pipeline integrates this data. Then we also have protein sequences from the UniProt database. And then we also can use some homology relationships to infer gene annotation as well. So for example, we can predict the genes in the chimpanzee by mapping the human cDNA and proteins to the chimp genome. Obviously, we know because of the close ancestral relationship of human and chimpanzee that the human sequences will align very closely to the chimp genome. So this is what the pipeline does. It takes the reference genome assembly from the archives, and then into the pipeline, we feed all of this evidence. So the nucleotide sequences, the protein sequences, the homology relationships. And this all goes into the pipeline, and then the pipeline aligns these sequences to the genome and uses it as evidence to make the gene predictions. The other type of annotation, the other methodology, as I mentioned, is the curators. So it's a group called Havana. You can see the name here. This is the group of Havana curators. They're actually based in Ensembl, so they're just in the offices down the corridor from us in Cambridge. And they're a group of curators that walk metaphorically along the genome, annotating the genes, using also nucleotide sequences, RNA-seq data sets, long-read transcriptomic data, intron data, poly-A-seq data, Oxford nanopore technology data, and also a range of publications and mass spec data as well. So they integrate a wider range of data in their annotation as well. So I'm guessing that you can already begin imagining the benefits and the disadvantages of performing the manual annotation. So the benefits is that the manual annotation is much more comprehensive, and you require less evidence. So the curators can look at the evidence, they can appraise the quality, they can look at the source, they can look at how it's aligning to the genome and integrate that all together and make a decision on the annotation. Whereas sometimes when you're using an automated pipeline, things get skipped over if there's not the required threshold of data to make an annotation. So it's this balance between thresholds and quality of underlying data that the manual curators can really dig into and make good annotation with. And the manual annotators are also really good at predicting these sorts of features and structures that are quite difficult to predict for the automated pipeline. So different UTR lengths, alternative splice sites, single exon transcripts, all these sorts of things. Obviously, the disadvantage is that it's a much slower and much more expensive process. And it happens on because of this, it happens on a much smaller scale. So this is why we only have the manual annotation for 4 species so far. When you're looking through Ensembl, what you're actually going to find sometimes is what we call golden transcripts. So whenever you see a golden gene or a golden transcript in Ensembl, like this, so it's just colored gold, it tells you that both the automated and the manual annotation methods have independently come up with the same annotation. So the same length, the same intron-extron structure, the same sequence. So you'll find golden transcripts for human, mouse, rat, and zebrafish, where the independent annotation methods agree with one another. So what we actually have in Ensembl is what's called the GenCode gene set. So whenever you're looking in Ensembl, the name of the gene set that you're looking at is called the GenCode gene set. So this is just the brand, or like the brand name or the term that's given to the genes that are produced by Ensembl. So if you're ever reading a paper or looking at a publication and it says the GenCode gene set was used in our annotation or in our analysis or something like that, then you've known that they've come to Ensembl and retrieved the Ensembl genes, which is also called the GenCode gene set. So the GenCode gene set is the merged set of Ensembl automatically annotated genes and Havana manually annotated genes. So as I've mentioned, when the automated and the manual methods are annotating genes, what they're actually doing is they're annotating transcripts. So we know that a single gene can have multiple transcripts, and these transcripts might be expressed at different levels, in different tissues, under different conditions. But you have a single gene with many different transcripts. Some might be more biologically relevant than others. Some might be only relevant at certain times in certain developmental stages. But what Ensembl does is it annotates all of the transcripts that we have evidence for from different sources. And then we leave it up to you, the scientists and the researchers that are looking at Ensembl, to look through the transcripts and try and work out which are the most relevant for your particular studies in your cases. However, what we appreciate is that sometimes it can be really difficult to try and take all of the different transcripts of a single gene and pick one or two that might be the most relevant for your study. So what we have done is we've developed a project called the MANE project, M-A-N-E, and this is a collaborative project with NCBI to annotate a single biologically relevant, clinically important, and highly expressed transcript for each protein coding gene that's agreed upon between Ensembl, but also the NCBI databases that are hosted in the US, in the United States. So MANE, M-A-N-E, stands for Matched Annotation from the NCBI and EBI project. So as I mentioned, the project aims to identify a single biologically relevant transcript for each gene that has 100% identity between Ensembl and NCBI. That's also biologically relevant and clinically important. So at the moment we have, this is an ongoing project. So MANE Select version 0.95 is the current version of the MANE project, where we have 95% of the human protein coding genes that have a MANE Select transcript annotated for it. So what we'll look at in the demonstration later on is how we can look at all of the transcripts of a single gene and try and find the MANE Select transcript and prioritize and filter the other ones in terms of relevance and importance. So this is what we're going to be doing in the demo later on. So the question that we're going to try and answer at some point in the demo is, which transcript should I use? So this is a screenshot of the Ensembl browser. We're going to come across a table like this in the demo. You can see that the transcripts are presented in a table like this. So the transcripts have got these IDs, but then we can find out information about the transcripts, like links to other databases, but we can also identify these flags as well. So we can see if the transcript is part of the MANE Select. It's been annotated as the MANE Select transcript. We can also see if a transcript has been annotated by the APRIS project. So the APRIS project is a collaborative project that's based in Barcelona in Spain, and they also are working on annotating a single, what they call principal isoform for each gene. So they look at protein structural information, functionally important residues and alignments to try and, again, like the MANE project, identify a principal isoform or a single transcript that's the most important for each gene. You can also see if a transcript is part of the GenCode basic gene set, which means that it's a full-length transcript with a start and a stop codon. It's not incomplete or fragmented annotation. And we can also see what we call the transcript support level. So TSL is a score of between 1 and 5 that we give to each transcript that tells us the quality of the underlying data. So a TSL score of 1 means that the underlying data is unambiguous, whereas a TSL score of 5 might mean that there's some ambiguity or some issues that are raised with the underlying data that's been used in the annotation. So these are the different things that we can look at, and we'll think about this in the demo later on as well. So up until now, I've been describing the annotation process and some of the associated data for the genes and the transcripts in the Ensembl project, so for the vertebrate species. Now, I want to just spend a few minutes thinking about the annotation in the Ensembl genomes project, so for the non-vertebrate species, because it's slightly different. So for the non-vertebrate species in Ensembl genomes, Ensembl does not perform the annotation ourselves. So the annotation is performed by other research groups and collaborative projects. However, they do use very similar methods. So for example, the Ensembl genomes annotation comes from these other projects and is imported into Ensembl, and they might have used an automated annotation pipeline similar to the Ensembl pipeline. They might have used some homology annotation. They might have a group of manual curators as well, just like Ensembl. And then they also might have an ab initio prediction pipeline looking at open reading frames within the sequence. So this is just an example of some of the different non-vertebrate species and the different databases and projects that are responsible for doing the annotation. So if you are studying plants, maybe you're looking at the breadwheat, Triticum aestivum, then the Earlham Institute is the group that is responsible for sequencing the genome, but also doing the gene annotation as well. So they have a mixture of automated and homology annotation methodology. Then you can look at something like Arabidopsis, which is a model organism again for plants. And the TARE project, they, as well as having the automated and the homology annotation, they also have some manual curators that are responsible for annotating the genes as well. For different species, there are different databases. So you have like POMBase for Schizosaccharomyces pombi, and then you also have FlyBase for Drosophila as well. So what we do is we directly import the genome sequences and the gene annotation directly from the ENA database. So ENA is, we've come across this before, it stands for European Nucleotide Archive. And as well as having the genome sequences, the researchers will have deposited the annotation as well. So you can see here, for example, this is a screenshot from ENA. This is for a bacteria for Staphylococcus aureus. So there's some researchers at Columbia University have sequenced the genome, and they also have the gene annotation as well. So Ensembl imports this genome and annotation and presents it in the in our web pages. So before we start the demo, I want to quickly revisit some of the things that we thought about in the previous module. So when you're looking at a gene, you can actually see, as we saw in the last demo, that the genes are represented as the individual transcripts. So these are all of the transcripts of the BRCA2 gene, for example. So the first thing to notice is that each of the transcripts is color coordinated. So we can see that this is a golden transcript at the top, and we know what the gold transcript is. It's a merged transcript. So we know that this one at the top has been independently annotated by the automated and the manual annotation methods that have come up with the same structure of the transcript. Then we have these other transcripts. So this is a red transcript underneath, for example. And whenever you see a red transcript, it means that it's a protein coding transcript. And you can see that in the description here as well, protein coding. Now, the golden transcript is also protein coding. So if it were not gold, it would be red. So the gold color just is more important, so that's laid on top of the red. Then we have what we call the non-coding transcripts, which are in blue. So these are transcripts that do not give rise to any functional protein products. So they might be nonsense-mediated decay transcripts or process transcripts or retained intron transcripts. If we take a look at one of these transcripts individually, we can see it's made up of these boxes and lines. So this is the representation of the transcript, and it's got these boxes and lines. So the boxes are the exons, and the lines are the introns. So you can see this is an intron here, and another intron, and another intron, and then these are the boxes are the exons. So wherever you see a filled in box like this, this is a coding exon. So the color, it's got block color inside it. It's a coding exon. Where you see this box here that's not filled in with any color, it's just blank, you can see that this is a non-coding exon. So for example, this portion of the exon at the end here, this is the 3' UTR. So it's still part of the exon sequence, but it's not coding. It's just the 3' UTR sequence. Good. And then finally, before the demo, I wanted to also mention to you about stable IDs. So when Ensembl is performing annotation, we assign all of the features with a stable identifier, a stable ID. So in Ensembl, it always starts ENSG for genes, and then it will be a unique 11-digit number that identifies that gene. And then we also have ENST for transcripts, ENSP for peptides, ENSE for exons, and ENSR for regulatory regions. So obviously, when we're using Ensembl and when we're talking about genes in the lab, we actually are more likely to use the gene names. So BRCA2 or ESPN, for example, or MYH9. So we often use the gene names, but we know that gene names are not consistent and they're not necessarily stable. So if you're studying a gene, it's much more important to make a note of the stable ID, because it means that if you come back to Ensembl in the future and there's a new synonym or a new gene name for your gene of interest, that might mean that you can't find it, but the stable ID will always be the same from one Ensembl release to the next. So making a note of the stable ID is a really important thing to do. For non-human species, there's a suffix in the ID. So for mouse, for example, it's ENS-MUS-G. For zebrafish, ENS-DARG for Danio Ririo. So all of the different species will have a 3-letter suffix in there. If you're studying a non-vertebrate species, so if you're studying a species in Ensembl genomes, because the annotation comes from the research groups and the collaborations themselves, sometimes they assign their own stable IDs. So they might not have this, they will have stable IDs, but they won't have this structure of ENSG, for example. Good. So what we're going to do now is we're going to move on and have a look at the genes and the transcripts in Ensembl. So we're going to search for a gene called UQCRQ, and we're going to explore the gene tab and all of its different transcripts. So the first thing I'm going to do is open up a new tab in my internet browser and go to www.ensembl.org. So I'm going to go back to the Ensembl homepage, and I'm going to search for my gene of interest. So this is UQCRQ. And I'm going to just click, well, firstly, I'll copy this and put it in the chat box so that everyone can copy and paste if you need to. And then you can click go. So when you search for your gene, you can see that in the search results, the human gene is listed at the top here. This is by default, the human gene is always listed if it's available. But you can also filter to search for your species of interest using the filters on the left-hand side if you want to. We won't use any filters. We're just going to click on the link for the human gene if, in this case, in our example, we're interested in the human gene. So you can see here that we've landed on the gene tab. So just as I mentioned to you in the previous demo, we're opening up these tabs in the blue bar for the different features that we're exploring. So now we're in the gene tab, and you can see we have a menu on the left-hand side, but it's different to the one we saw before, because we're in the gene tab, so we have different data available relating to our gene of interest. However, we have opened up a location tab here by default. So what you can actually do is you can flick between the tabs to see the different views. So if you click on the location tab, you'll see this is the page that we saw before, but this is the genomic location on chromosome 5 where the UQ-CRQ gene is found. So you can obviously come here and explore the UQ-CRQ gene and the tracks, or you can go back to the gene tab and explore the gene through these pages as well. So we're obviously going to explore the gene tab now, but this is just showing you how you can navigate between the tabs to explore all the data that's available. So we've landed on the summary page. You can see summary at the top here that's highlighted. So here's our summary. So we've got the gene name and the stable ID. Then we've got a description and some of the other synonyms for this gene, the location and the strand. And then we have a blue button that shows us, is asking us if we want to display the transcript table. So if we click on this button, we're going to open up the transcript table for this gene. So this is similar. Well, this is the table that we saw in the presentation that shows us all of the transcripts of the UQ-CRQ gene. So you can see the transcript ID, the name, the length of the transcript in base pairs, and if it's a protein coding transcript, you can see the length of the protein. We've got the biotype, so we've got protein coding, you can see, or we've got these blue transcripts which are non-coding, and if you hover your mouse, you can see a short description of the biotype of the transcripts. Then we also have links to other databases. So we have links to the CCDS, the UniProt, and the RefSeq databases as well, if there are corresponding entries. So if you want to see, for example, the protein that this transcript encodes, so this is the protein entry from UniProt, you can click on this link and it will take you out to the UniProt databases. So here we can see the UniProt databases that have information about the protein structure and function. Obviously, Ensembl is a database about genes and genomes. So where you want to find information about the proteins, you need to go out to a database like UniProt. But you can do that from Ensembl, like I just showed you. You also have links to NCBI as well here. So this is the RefSeq transcript from NCBI. So you can click here, and this will take you out to the NCBI pages as well. So this is where Ensembl is really good. What you can actually do is you can begin cross-referencing with other databases and trying to build up a good idea about this gene and its transcripts and all of the information that's available for it. So over on the right-hand side of the table, we have this column called flags. So this is where you can begin looking at the transcripts one by one and trying to prioritize and work out if there's a single transcript that's the most important for your studies. So we can see that this top transcript is the main select transcript. And if you're studying human data and you're looking at a gene, I would always say that if a gene has a main select transcript, this is the one that you should investigate firstly. So this is the transcript that's agreed upon and biologically relevant and highly expressed. So this is the one that you should begin investigating, with knowing that there are other transcripts which might also be important in your studies as well. Then you can see that this transcript is also part of the GenCode basic set. So it's full length. It's not 5' or 3' incomplete. And then we also have the APRIS annotation. So this transcript is also the principal isoform as predicted by the APRIS group in Barcelona in Spain. And then we have a TSL score of 1. So if I was to be looking at this gene, I would prioritize this transcript at the top here as the most important transcript for my study. And then I might also look into these other 2 protein coding transcripts. They might be relevant in specific conditions. Good. Further down, we can see again a summary, so just some links to other databases. And then at the bottom, we have also it's a graphical representation of all the transcripts. So you can see the boxes and the lines that represent the exons and the introns here. Good. So what I want to do is spend a little bit of time having a look at the menu on the left-hand side. So this is just the summary page. So now we're going to explore some of the different options here in the menu on the left. So firstly, I'm going to click on sequence. And I'm going to hide this transcript table just because it appears on every page. So I'm just going to collapse it so it's easier to see. So what we can see here is what we call this is the genomic sequence where the UQ-CRQ gene is found. So there's a number of things that you can notice about this sequence already. I already have some annotation on this sequence. So what you can actually do is you can click on configure this page now, and you can edit and change the sequence, change the formatting of this sequence in this view. So by default, you'll have 600 base pairs upstream and downstream, but you can change this so that you're just seeing a shorter upstream sequence, for example. You can also choose to look at variants on the sequence if you want to. And you can also add line numbering if you want to as well. So these are lots of different options for the formatting of the sequence. When you're happy, you can save and close using the tick in the top right-hand corner. So you can see that the page is just reloading. So I already have the 200 base pairs and then the line numbering. When you're looking at the legend, you can see that the variants are loading, but you can already see the exons annotated directly on the sequence. So this is the FASTA sequence, and you can see that all of the exons. Maybe I'm just going to remove the variation data because I think it makes it a bit more difficult to see. So when you look here, you can see that all of the exons in this region are highlighted with this peach highlighting color here. However, only the UQ-CRQ exons have got the red lettering. So what that means is that this here is a UQ-CRQ exon, but upstream here, we can see that we have these exons that have got black lettering. So these are exons of neighboring genes. There's a number of things you can do with this sequence. So as you saw, you can click and drag and highlight a portion of the sequence. And when you let go of your mouse, you're asked if you want to blast the sequence. So you can blast some selected sequence if you want to, or you can download the sequence as well. So if you want to download, you can click on the blue button at the top here, and that will open up a pop-up window that allows you to download the sequence. So there's 2 different formats available. There's FASTA format and RTF format. So FASTA format is just the plain text file format that you might want to use to input into other tools, primer designing tools or platforms, for example. And you can choose if you want the genomic sequence or the coding sequence or the cDNA sequence. So you choose what type of sequence you want from the options, and then you can click download. The other format is called RTF. So this stands for rich text format. And this is not the plain text sequence like FASTA. This is the annotated sequence. So if you download this file format, you'll get the sequence, but you'll have all of the annotation on the sequence, like the the red lettering for the exons and the peach highlighting and the variants if you choose to see them as well. So this is not so useful for using as an input into other tools, but it's quite nice to visualize the sequence and all of the features that are annotated there as well. So these are the options that you can select for your annotation, and then you would click download. So I'm not going to download anything now, but you can obviously play around with these options and download the sequences of your gene that you're interested in. Good. The next thing I'm going to do is introduce you to the gene ontologies. So just by using your thumbs up icon or thumbs down icon in Zoom, can you tell me who has heard of gene ontologies before? Is it a concept that people are familiar with, or is it something that you've never heard of before? So if you've heard of gene ontology, then have a thumbs up. If you've not heard of gene ontology before, then maybe either do a thumbs down or type no in the chat box. Okay, so some people have heard of gene ontologies and some people haven't. So I just wanted to spend just a couple of minutes then explaining gene ontologies before we look at it in the browser. So gene ontology stands for, well, the acronym GO stands for gene ontology. So I want to try and explain gene ontology with an example. So we, as scientists, and even as humans with human language, we can have multiple terms for the same thing. So if we were met at a conference, for example, I might have a poster and I talk to you about my gene that was involved in innate immunity. And you might think that's really interesting. Then maybe you go home back to India after the conference and you talk to your lab mates and you say, I met this guy called Ben. He had this poster and it was about a gene that's involved in non-specific immunity. Now, between all three of us, we know that we're actually talking about the same biological process, but we're actually using different terms to describe the same thing. And we can also be really specific as well. So I might talk to you about my gene that's involved in the complement cascade, which, because you're a scientist and you can infer from languages, you know that that's part of the innate immune function, but I've never actually used those terms to describe it. I've just used the term complement cascade. So basically, human language can be really ambiguous. We can have different terms. We can, as listeners and speakers, we can infer different things from the words that we use. However, computers are not good at interpreting language. So what we have done, and when I say we, I mean the scientific community. So what the scientific community has done, there's a project called the Gene Ontology Project, and they've developed a standardized vocabulary of terms that describes gene function. So all genes that are implicated in the innate immune response will be tagged with this specific gene ontology ID. So what this means now is that everything is standardized and very searchable. So now we can look at a gene and we can find all of the gene ontology terms that are linked to it, and they're going to have a very standard definition and structure. So thinking about that, we can now go back to our example and we can click on the ontology section in the menu on the left. So if we click on biological process, we can begin to see the biological processes that the UQ-CRQ gene is implicated in, or that it's involved in. So we can see that the UQ-CRQ gene is involved in subthalamus development, hippocampus development, hypothalamus development, midbrain development. So it's all involved in this sort of brain tissue development. It's also involved in this mitochondrial electron transport process as well. So what we're beginning to do now is build up an idea of the known function of this gene. This is the evidence and the annotation source here you can see in the table. If you click on the cellular component, section of the ontologies menu, you can now find a list of gene ontology terms that describes the location within the cell that this gene is involved in. So the protein, the UQCRQ protein, is found in the mitochondrion, the mitochondrial inner membrane, and even more specifically, the respiratory chain complex 3. So this is the location within the cell that this gene is involved in. So again, helping you build up an idea of the structure and function of the gene and the protein. Good. Okay. So the final, the penultimate thing I want to show you is this section called gene expression in the menu on the left-hand side. So if you click on gene expression, you'll be able to find a page that shows you the tissue-specific expression patterns of the UQCRQ gene. So over on the left-hand side, we have a diagram. So we've got male tissues, female tissues, and brain tissues. And then over on the right-hand side, we have a table that shows us the expression levels of the UQCRQ gene, identified in a number of different projects in a number of different tissues. So we have the projects on the x-axis here, and then all the different tissues on the y-axis here. So for example, we can see that this Phantom 5 project identified that in the cerebellum, the UQCRQ gene is expressed, the expression level is 66 TPM. So the TPM stands for transcripts per million. It's a sort of a measure of expression levels. So you can see this is quite a dark blue color. As you scroll down, you might see that there's some light blue expression patterns. So this might be a tissue where this gene is expressed at a lower level. So the dark blue means high expression, and then the light blue means lower expression. Where there's gray, it means it's below cutoff. And obviously, the white tells us that there's that tissue was not analyzed in this experiment. So what you can do is you can filter this table. You can drag and zoom into the table, so you can begin looking at specific tissues and looking at the data that's really interesting to you. Good. The final thing that I want to show you on the Gene tab is at the bottom here. It's called External References. So if you click on External References, you'll find a table that shows you links to all of the external databases that also have information about the UQCRQ gene. So you can see that we have a link to Expression Atlas. So this is a database also based in Cambridge here in the UK that has information about the gene, the tissue-specific expression patterns. And this is actually the source of the data that we were looking at on the previous page. Then we also have these databases called MIM-Gene and MIM-Morbid that have information about phenotype and disease association. So this is a database that you can look to see if your gene is involved in any phenotypes and diseases. Then we've got NCBI as well. That's quite a well-known database that you can explore as well. And then Reactome and Wikigene. So these are all links that you can click on to go out to the other databases and explore. Good. So in the final few minutes of this demo, before I set you off on the exercises, I just want to quickly explore the transcript tab. So at the moment, we've been looking at the gene tab by itself. If we open the transcripts table by clicking on the blue button at the top here, we can actually investigate a single transcript in more detail. So if I open the table and click on the link here in the table for the transcripts, that's the main select transcript, I can click here and this will open up the transcripts tab. So you can see now I've got the transcript tab at the top here, and the menu on the left hand side again has changed because we now have information and data relating specifically to this transcript of the UQCRQ gene. So we've landed on a summary page. You can see a summary of the transcript at the top here. You can see the transcript table, obviously showing you the other transcripts still. And then the summary at the bottom, it just shows you the structure. So you can see the exon and the intron structure. And then you can see some information about the length of the transcript and the number of protein amino acids residues in its translated protein. Good. So what I want to do now is explore the sequence of this transcript. So I'm firstly going to click on exons in the menu on the left-hand side. So what you can see on, what I'm going to do firstly is hide the table again, the transcript table. And if we scroll down, we can see all of the sequences of all of the exons of this transcript. And it's divided up into this table here. So we can see here, the 5' upstream sequence is in green. So this is just the upstream sequence. Then the first exon, you can see it's labeled as number 1 in the table. These are the coordinates. This is the length, and this is the sequence. So you can see that this you can see that this sequence is colored in this red color, which means that this is UTR. So this is non-translated sequence. It's just part of the 5' UTR. Then we have intron 1 to 2. So this is the intronic sequence between exon 1 and 2 that's just listed in gray. Then we have exon 2, and again, we can see that the sequence this time, we've got this red sequence, which is part of the UTR, but then we also have now blue sequence. So we can see from the legend at the top that the blue is translated sequence. So here we can see this is the ATG, this is the start codon, and then this is the rest of the translated sequence of this exon. Then we have another intron here, and then we have exon 3. Again, the blue translated sequence, finishing with the TGA. This is the stop codon. And then finally, we have the 3' UTR here in the orange-red color. You can do exactly the same things as we saw before. So you can click and drag if you want to blast the sequence. You can download the sequence using the button at the top here. And you can configure the sequence using the configure this page button here. So if you want to configure the page, you can choose to show or hide the full intronic sequence. You can add line numbering if you want to. And you can even add variation data if you want to as well. So there's lots of different options. And again, I'm just using some of these as an example, but you'll obviously want to think about what sequence do you want to see and what annotation is important for you to view. Now you can see the variants are also loaded on the sequence. The next thing I'm going to click on is the cDNA sequence in the menu on the left. And this shows you the cDNA sequence of the transcript. So here we have a sequence that's split into each line with 3 separate rows. So in line 1, we have the cDNA sequence at the top here. Then we have the coding sequence on row 2. Then we have the protein sequence on row 3. So the cDNA sequence firstly starts here. This is the UTR in dark yellow. And then we move into the coding sequence. So we have ATG is our first triplet codon that encodes a methionine. Then our next triplet codon is GGC, which encodes the glycine. Then CGC encodes the arginine. So you can see that the triplet codons are sort of arranged with this white and light yellow highlighting. And you can see the corresponding amino acid here as well. Good. So the final thing I want to show you is in the menu on the left-hand side called the protein summary. So if you click on protein summary in the menu on the left-hand side, you'll be able to see a depiction of the protein is in a linear form with all of the different predicted motifs and domains that exist in the length of the protein. So in the image, you can see this is the protein. So this is the N-terminal tail on the left, and this is the C-terminus over on the right-hand side down here. And then underneath, you can see all of these different databases and prediction methods that have predicted the different domains and motifs that exist within the peptide structure. So because this is quite a short protein, that's just a single globular protein, there's just one predicted domain called the cytochrome BC1 complex. So superfamily, PFAM, Panther, Gene3D, these have all predicted the existence of this particular protein domain across the whole length of this protein. So obviously, if you're looking at a longer protein, you might find that there are different domains that exist in the M-terminus and the C-terminus, and you can begin to get an idea of how the protein folds and the different domains that are and motifs that are involved in the protein structure and function as well. Good. Okay. So that brings me to the end of the demo. We just have around about 10 or 15 minutes to have a go at the exercises for this module. So if we go back to the training materials, we can click on the genes and transcripts module. And again, you can open up the exercises. So we have a section for exploring a human gene, and then an exercise for finding a gene associated with a phenotype. So I haven't specifically shown you how to do this, but you might be able to use the instructions within the exercise to search and find the gene associated with a phenotype. Then there's an example with a mouse gene, and then we also have a plant gene at the bottom here as well, Arabidopsis thaliana. So please spend the next 10 minutes or so having a go at those exercises. And then at 5 o'clock, I'm just going to come back, bring us all back together to see if there are any final questions. And then we're going to wrap up and ready for tomorrow. So before I set you off, are there any questions about the genes and transcripts pages? Good. So if there are no questions, please have a go at those exercises. Louise and I, again, will be on hand. If you have any questions, please raise your hand or put them in the chat box or the Living Doc. Hello, everyone. It's just nearly 5 o'clock there in India, so in Indian Standard Time. So it's the scheduled end of our workshop session today. I hope you found it useful. We'll be back again tomorrow for the next module. So Louise will be delivering the variation and comparative genomics modules tomorrow. We do have a couple of minutes if you want to have any final questions. If not, then please feel free to have a go at the exercises in your own time before tomorrow's session. And if you do have any questions, then you can put them in the living document, and then Louise and I will try and answer them before tomorrow's session if you have them in before the next session begins.
Thank you for today's session. It was wonderful. I was actually trying to find out the answers, taking screenshots and fixing up, pasting it in a Word document. So far, it will be there. Thank you so much.
That's great. I'm glad you found it useful. Yeah, hopefully, yeah, as I said, the exercises are not a test. Like it's great that you're sort of like using it to, you know, make a sort of a reference document and like document your answers, but there's no need to unless you think you're going to find it useful. Yeah, the exercises are just a starting point. You can obviously go off and explore in so many different directions.
Thank you, Ben, for the wonderful lecture. So tomorrow in the same Zoom link, we'll do the program.
Yes, so it's the same time, same Zoom link tomorrow.
Okay, if you have any question for the final time, you can ask now, and then we will meet tomorrow.
Well, sir, I have a question.
Of course.
Can I put in the question?
Yes, you can ask the question. If you want to ask the question now, you can, or you can.
What I was asking is the exercises are available even after the session?
Yes. So so the materials, the link that I shared in the Zoom chat box, I can put it there again for you. The materials are all
of the exercises we are not able to complete by this time, so we can do it later on also.
Yes, yeah, and even the screenshots from the demos, everything will still be there after the course, so you can come back and continue your exercises, or you can just come back and use it as a reference if you want to remember what we were doing and find the right section.
Thank you. Thank you, sir.
No problem at all. Okay, perfect. So I think if there's no questions then we can finish the call for today. I hope you all have a nice evening, and we will see you tomorrow.